guys, I just want to give us our notes for this unit. This is the first part of our notes that we will be having. We'll have another packet to do probably in a week or two. The first part's going to focus on everything that we've been doing with the cell cycle, mitosis, and all of that. So everything in here should be fairly familiar with you. All the terms is stuff that you've done for vocabulary, for homework, and all the cell cycle stuff is everything we've been doing thus far. And we'll continue doing that this week. So the first thing let's do is let's look at what you should be looking at in your Google Classroom. You should have a copy of the notes, short version it says. Believe it or not, this is the short version. Lots of steps have been removed from the notes because we no longer teach all the steps of mitosis. Lucky kids. So anyway, it should be, I think it's 3 pages total. So three Google Doc pages. Scroll through, there you go. It ends in slide 20. So make sure you either print it out and write on it and upload that, or write directly, type directly onto the Google Doc. Either which way is fine. All right, so let's do the notes. Okay, it's fairly short, so hopefully it won't take us all day. It won't. All right, so structure, function, and growth of cells. That's what we're looking at today. I want you to think about how organisms go from a single cell to these multicellular objects. They are not just single cells getting much, much bigger. Obviously, we know that, right? We're actually getting multiples of different cell types to make leaves, to make xylem, to make phloem, to make tree bark, okay? Or to make muscles and hearts and bloods and bones and teeth and all that. They're different types of cells. So, because we need to have different types of cells, and I know we've seen this last week when we watched the kidney video, we saw that cells do divide and they multiply. So they divide and become more numerous is what that means. Kind of just like in math class, we do division and multiplication. You're going to do it in bio too. All right, they have to do this because cells have to be able to do specialized functions. So that means they get specialized or differentiated and they become different cells that the organism needs. So depending on what kind of organism you have, if you have a mammal, you have blood cells. If you have a plant, you have plant cells, and they have leaf cells versus the interior, like the woody xylem and phloem, and those pith of the tree. Those are different cell types. Okay, here's a stomata, it's a neuron. We have lots and lots of different cell types. So we have red blood cells, plant cells, guard cells, neurons, skin cells, white blood cells, and the sperm and smooth muscle cells right here. All right, there's 2 ways that we have cells that are able to reproduce. The first way is asexual reproduction, and this is where you have a single parent producing an offspring. There's no mixing of DNA, okay? The offspring that are produced are genetically identical to the single parent cell. We have an example right here of bacteria, and bacteria really produced by a more specific process called binary fission, and you have one cell, lots and lots. See how the DNA inside here is duplicated? There's way more DNA, and then we have it separating out, and then at the end you have 2 identical, genetically identical daughter cells, and that's what these guys are called here, is daughter cells. Sexual reproduction, on the other hand, is a fusion of 2 sex cells. You get one from your mom, one from your dad, one from the female, one from the male organism, and they combine to become a mixture. And the offspring inherit genetic information from both of your parents. This occurs for most animals and plants. I have a nice family picture here of the royal family before you know, Meghan and Harry departed the family and moved to California. Okay, before their baby was born, Archie. Just trying to remember his name. All right, next. We're kind of flying through this, so you feel free to pause as we go. Sorry. On your packet, you need to fill in the table that compares asexual reproduction and sexual reproduction. Asexual reproduction produces genetically identical offspring, is very quick, and produces a lot of offspring obviously in a very quick amount of time. Sexual reproduction will increase the genetic diversity, so you get a more diverse offspring, and that will help ensure that the survival of the species the species survives if and when there's changes to the environment, right? So if you have a variety of fur colors, if the Arctic suddenly melts away and you're left with a grayish surface, darker colored organisms are going to survive easier than the white colored ones. Just like if you have glaciers developing in areas, white or light colored ones are going to survive better than the darker ones. Okay? So feel free to pause and fill that in. And it won't go. Not sure why. Okay. Why cells need to divide. So if a cell continues to get very big all by itself, it's going to have a harder time efficiently moving waste products out, because all these products have, or all these processes that are occurring in the cell produce a waste product. And remember, for photosynthesis, that waste product was oxygen. So if you have chloroplast in the center of it, the bigger the cell gets, the harder it is for that oxygen to escape. It has more cytoplasm that it has to go through. It's also going to take longer for the items, the oxygen, or the carbon dioxide, to get into the chloroplast where it's needed, because there's more cytoplasm that it has to go through, and it has to navigate through all the different other things that are in there too, the other organelles. So it makes sense that you want to keep these guys, or these cells, at a reasonable size so that material can move in and out as needed. As the cells get bigger, though, their volume is going to increase faster than their surface area, and that is also contributing to why these materials are harder to move in and out, because it has more volume that it has to go through. So that's 2 reasons why cells definitely need to divide to keep their size consistent. All right, cell division. Before a cell grows too large, it divides into 2 new daughter cells, and this is through the process of mitosis, and this is what we talked about last week. Number 1, that's important because it reduces the cell volume. That will increase the ratio of surface area to volume for each daughter cell. That used to be a test question. We're not having a test, so it's emphasized here because it's something that's emphasized on a test. Daughter cells, DNA, is identical to the parent cell, which we saw in our cell cycle one. There was no introduction of new DNA. Everything was the same. Chromosomes are how genetic information is passed from one generation to the next. It is put into chromosomes because it's organized. It makes it easier for cells to make copies of their DNA when we get to the next part of our unit, which is that copying of DNA. And it's really easy to divide a chromosome in half, and you know you have, if you've done it correctly, if your chromosomes are not mutated, you have equal amounts of genetic information on both strands. So it's important that they be organized in a way that you can easily and efficiently check for errors, check to make sure that there's no loss of genetic information, and distribute it equally and quickly. It's a very efficient means to do that. All right, here's our structure of a duplicated chromosome. A chromosome is just 1/2. So this is a chromatid, a sister chromatid. That's a chromosome. And then another sister chromatid. So this is actually 2 chromosomes right here. And then the centromere is what holds those 2 chromosomes together. Eventually they'll get pulled apart during one of the steps that we're not going to worry about naming. Prokaryotic chromosomes are not structured the way we just saw. In fact, theirs is usually singular. And it's kind of like how it's pictured on here, like a loop, okay? And there's a different way that it's replicated than the chromosomes that are in eukaryotes. The chromosomes are also, because it's a prokaryote, not in a nucleus. It's just in the cytoplasm. So it's just got DNA floating around in there, okay? But your eukaryotic chromosome, this is how it boils down if you start looking at it. So you have your duplicated chromosome here. Where's my mouse? There it is here. So 1 chromosome is here, a sister chromatid, and it's all coiled tightly in there. So it's coiled upon coiled, because you can see this is coiled again, upon another coil, and eventually you uncoil it enough and you get the structure of the DNA double helix. Okay, and this is all tight. This is how you can pack so much into a chromosome into a single cell. And having that information in there, okay? That was not supposed to happen. Okay. here's our eukaryotic cell cycle. Hopefully by now we're very familiar with this. So our G1 phase, we have cell growth. There's a checkpoint in between G1 and S phase before our DNA is replicated, right? Then there's another checkpoint after DNA has been replicated before it enters the M phase, right? And it makes sure that the DNA has replicated correctly. There's enough genetic information, all that good stuff. There's more growth here. And then we enter the M phase, which is your cell division phase. This whole other part is interphase. We drew this out last week. Hopefully all of this is familiar to you. Then we have mitosis, which divides the nucleus, and cytokinesis, which divides the cell, or cytoplasm. And I talked to you guys about, with the animal cells, the cleavage that occurs. It's called a cleavage furrow. Before those two cells, it's the membrane that pinches together and breaks apart. So you get your two cells. Something similar happens, but under a different name for plant cells. And I'll get to that in a moment. So our two phases, mitosis, division of cell nucleus, and cytokinesis. Just remember cyto and cyto. Cytokinesis is the division of cytoplasm. Which you said again. So here's that cleavage furrow I was talking about in animal cells. And then in plant cells, they have cell walls instead of cell membranes. Well, they have cell membranes within the cell walls. But that wall is a harder, firmer material. So instead of a cleavage furrow forming, you get a cell plate. And this kind of forms along the middle. And once it's formed all the way across, the cells split apart. Okay. Now, cyclins. We talked about these quite a bit last week with our cancer unit. It's a family of proteins that regulate the timing of the cell cycle in eukaryotic cells. So there's two types of cyclins. We have internal and external. And much like their names imply, internal is going to be responding to stimuli inside the cell versus the external ones that respond to events that are happening outside the cell. So the main example that you can have here is growth factors. So our bodies, like humans and other mammals, produce growth hormones, and those speed up cell division and can cause, that's when kids go through growth spurts. That's their growth hormones have triggered cell division and the cell cycle to continue to go at a more rapid rate. It also is very helpful when you have a cut. Things get triggered and the cell cycle gets initiated to go more rapidly and your wound will heal. That does slow down as you get older. If you look at my husband's grandpa when he was in his 90s, a fall and a cut took months to heal versus you guys, a cut takes a couple of weeks and it's gone. So, all right. The next thing we're going to talk about is stem cells. So we talked about eukaryotic and prokaryotic cells and that eukaryotic cells were specialized, right? Stem cells are kind of cool because they are not specialized, but they are able to reproduce very quickly, which is beneficial because we want to be able to take advantage of the fact that these cells have no differentiation. So they are capable of rapidly dividing and of self-renewal. They are unspecialized, meaning not differentiated yet. And that means any type of cell can develop from it. It doesn't have to be a bone cell going to a bone cell. It can be anything, okay? And they show that here, that this can be a macrophage, muscle, neuron, blastocyst, skin cell. I mean, there's all sorts of things that these stem cells could eventually become. So stem cell research is really revolutionary, I guess would be the best way to describe it. It's controversial, of course, because depending on where stem cells are harvested from, but it doesn't have to be from controversial sources. Let me put it to you that way. Okay, so stem cells offer the potential benefit of using undifferentiated cells to repair or replace badly damaged cells and tissues. Already, right now, we use these for bone marrow transplants. Hopefully, at some point down the road, we will be able to build new organs because organ shortages are huge, and being able to build a new kidney or a new lung or a new pancreas or whatever you need would be tremendously helpful. I don't know if we'll be able to do that anytime soon. I know there's lots of progress in this department and using stem cells in 3D printers even. Lots and lots of options coming. Hopefully we'll figure that out down the road. Okay? All right, so I think that was it. I don't think there's anything else. That is correct. Short notes packet this week. Please make sure you copy this all down and turn it in by Monday, the 25th. Next Monday, the 25th. By midnight in Google Classroom for me. If you have any questions, please bring them to class with you so that we can go over them. And that's it. Have a great rest of your day. See you in class.